This is the renal physiology tutorial presented by the S. Charles A.B. with reference Color Atlas of Physiology by Agamemnon Despopoulos and Stephen Silbernack. Hello, in this tutorial we are going to see the kidney, salt and water balance of the renal system. To start with, we are going to visualize a brief anatomy of the kidney. So now, to visualize a brief anatomy of the kidney, first you need to know that the kidney consists of a cortex, an outer medulla, and an inner medulla, which are all these portions. At level of the cortex, you're going to have the different blood vessels. And with the blood vessels, you have the afferent arterioles, which are going to enter to form the first capillaries, which are going to be the glomerulus. So, and when visualizing much more at the level of the cortex, you see that you're going to have an afferent arteriole and then an efferent arteriole, which are going to form the second capillary system, which are the peritubular capillaries. Another capillary system is also formed as a secondary capillary system, which is going to be called the vasa recta. But the difference between peritubular capillaries and vasa recta is that the vasa recta are much more long and parallel. So we have the interlobular artery here. These are the arteries involved here. We have the glomerulus here. Here is going to be the outer strip at this position. We have the outer strip of the medulla, and here you are going to have the inner strip of the medulla. So on the outer medulla, you have the outer strip and the inner strip. So now, after visualizing the blood vessels, you need to visualize now the different nephrons which are going to be involved with the renal system at level of the renal cortex. You have the cortical nephron. They are much more located in the cortex, and they have their lobe of inlay only descending and ending at the level of the inner strip of the outer medulla. So you have this descending, you see this portion, this thick descending limb. The thick descending limb is going to end at the outer strip of the outer medulla, while the thin descending limb goes far to the inner strip here and stops here, and continues the thin ascending limb to form the ductus, the distal convoluted tubules, and then enter into the collecting duct. We now have the juxtamedullary nephron on this side. On the juxtamedullary nephron, they have their lobe of Henle. The thick descending limb is going to stop at the level of the inner strip of the medulla, while here for cortical we're stopping at the level of outer strip of the medulla, of the outer medulla. So now it's going to stop at the inner strip of the outer medulla and then continues downward into the inner medulla. As it continues downwards inner medulla, it then goes up lobe of Henle, then produce, you see this portion here is going to be the distal part of the lobe of Henle, or the proximal part of the distal convoluted tubules. So you see that the proximal part of distal convoluted tubules start at this position here, at the outer strip of the outer medulla, and you're going to have these other portions here, and lastly it's over. It's going to enter now into your collecting duct. So the collecting duct is going to move from the cortex, or the cortices, it's going to move from the cortex and then it's going to enter into the medulla to form the different papilla. So we are going to have to all the collecting ducts are going to bind to each other to form the minor calyxes, then the major calyxes. Then what they are going to all enter into the renal pelvis, and then into the ureter. So this diagram here is produced due to a section across the kidney as such here. So now let's visualize the glomerulus. So the glomerulus and the Bowman capsule, we need to know that anytime filtration is to occur inside the kidneys, is going to pass through is going to be due to the glomerulus and the Bowman capsule. So now the glomerulus are going to be the capillaries, the first capillary system, which is going to be used in filtration of blood. So now you need to know that the glomerulus has a compacted capillary only in one tubule as such, only in one cup-like shaped structure, and that cup-like shaped structure which is encompassing the glomerulus is going to be called the Bowman capsule. Inside the Bowman capsule, we are going to have the capsular space, and here are the glomerular capillaries. Here is the glomerulus, all this bulk, and here is a Bowman capsule. The Bowman capsule consists of two main areas I'm going to see in the histology, which are the visceral and the parietal area. But the visceral layer of the Bowman capsule is the one which is involved with filtration. So now let's visualize the slit of filtration, the filtration membrane. The filtration membrane is going to be made up of three main layers. You have the endothelium of the blood. After the endothelium of the blood, you have the basal lamina plus the basement membrane of the endothelium and the basement membrane of the podocyte. So that's the second layer. After the basement membrane, then how you have the filtration slit. The filtration slit is due to the space which is formed between different pedicels of the podocyte. The podocytes are these cells here, and these podocytes have the foot. They have their different foot that they give to each other, and then their foot are going to have the small pedicels here, which are going to interjoin to form the filtration slit. And it's from that filtration slit that you can have filtration of heavy substances here. So you have a slit membrane, you have pores. You see that the pores here are going to be 5 nanometers, while the pores at the level of endothelium are 50 to 100 nanometers. So the more we are going down towards the Bowman's capsule, the more the space is reducing, and the less the larger protein structure, larger structures are going to be filtered through the filtration membrane. So now let's visualize a renal circulation. So now the renal circulation, we need to know that it's going to first start with the renal circulation. First start with the renal arteries. After the renal arteries, we have next the segmental arteries. After the segmental arteries, we have next the arcuate arteries. We have the renal arteries, the segmental arteries, then the lobar arteries. After the lobar arteries, we have the lobular arteries. After the lobular arteries, we have the arcuate arteries. After the arcuate arteries, we have the afferent arterioles. After the afferent arterioles, we have the efferent, we have the glomerulus, then the efferent arteriole. After the efferent arteriole, we have the peritubular capillaries. Then we have all the venules, which are going to have the respective names of the arteries which were given initially. So now, in this case, you have the blood vessels at the level of the cortex and the medulla. So the blood vessels mainly involved with the cortical nephron are going to be the second capillary system in mainly involve the cortical nephron are going to be the peritubular capillaries, while the second capillary system mainly involve the juxtaglomerular nephron are mainly going to be the vasa recta. So that's the main difference that we need to point out. So now, in this diagram here, this diagram is only a brief representation of this diagram. So let's go immediately to this diagram. This diagram here shows you how the kidney is capable of regulating its renal blood flow and the glomerular filtration rate. A point to note is that the glomerular filtration rate and the renal blood flow should always be kept constant in the kidney. So the kidney, no matter how you have the blood volume in the body, the blood pressure in the body, the kidney tries to maintain the glomerular filtration rate to be 18 liters, 180 liters per day, and it's always trying to maintain the renal blood flow to be having a value of 3.5 milliliters per minute per gram of tissues. So, and that can correspond to 5 liters. That can correspond to something like 2.5 liters of blood passing into the renal vessels. So all this should be maintained constant inside the body. So now the point to note is that, so after having this point to note, you need to know the margin in which the kidney can have its auto-regulation. So the auto-regulation is going to happen when the mean blood pressure is going to be low, and also when the mean blood pressure is going to be high. So normal blood pressure, the normal mean blood pressure is at the level of 90 to 92 millimeters of mercury. So the normal mean arterial blood pressure is going to be 90 to 92 millimeters of mercury. So now normally when the blood pressure decreases to the level of 80 millimeters of mercury, the renal system is capable of doing an auto-regulation where it's going to dilate the arterial blood vessels such that much more blood can flow inside the renal arteries, so maintaining the renal blood flow constant. Again, if you have a pressure which is higher, going to the level of a maximum value of 180 millimeters of mercury, you see that range from 90 to 180 millimeters of mercury, that's high blood pressure that the renal system can regulate. So what will happen is that there will be an auto constriction of the renal blood vessels in order to make sure that the renal blood flow should be kept constant. And again, you need to know that glomerular filtration rate is directly proportional to renal blood flow. The formula for glomerular filtration rate is a constant times the the net filtration pressure, and it's a constant. The constant is going to depend on the renal blood flow and the filtration membrane capability. And also the net filtration pressure will be equal to the total pressure inside the glomerulus minus the total pressure inside the Bowman capsule. Total pressure inside the glomerulus is the hydrostatic pressure inside the glomerulus minus the oncotic pressure inside the glomerulus. All this will be subtracted from the pressure inside the Bowman capsule, which is the hydrostatic pressure. The pressure inside the Bowman capsule will be equal to the hydrostatic pressure inside the Bowman capsule minus the oncotic pressure inside the Bowman capsule. So we have this. So now this diagram is going to show us the inulin clearance. So in this diagram, you see that inulin clearance equals to glomerular filtration rate. But before we go into this formula, we need to know what's the definition of clearance and what's the definition of glomerular filtration rate. So the clearance, let's first start with the definition of clearance. To define clearance, clearance is defined as the ability of the kidney to remove clearance is defined as the volume of plasma required to remove completely a substance from blood per unit time. So that's the definition of clearance. Clearance is defined as the volume of plasma required to remove. So there are three main statements involved with clearance. We have volume of plasma, first segment. Second segment is required to remove completely a substance from blood, that's the second segment. And the third segment is per unit time. So now if you have this, you can now say you can use the second statement as required to remove completely a substance from blood. That statement tells you that the amount of the substance excreted will be similar to the amount of the substance which was present in blood. So you can say N in blood will be equal to N which will be excreted inside urine. So after having all intersections, we know that the number of moles is equal to molarity times volume. So we are going to have on each side the molarity and the volume. Now since we are involved now with per unit time, this is why the volume will be in form of rates. Here the rate of filtration will be the glomerular filtration rate with respect to the blood on this side, and with respect to urine on this side is going to be the VU dot here, as you can see here. So when you are going to do this, you are going to find that the glomerular filtration rate or the clearance, which is equal to the clearance of inulin, is equal to U of inulin over P of inulin times VU. So we are going to have this. So in case of inulin, we need to know that no inulin is reabsorbed into blood and no inulin is going to be secreted inside the in the filtrate. So we realize that everything that is cleared is what was present initially in blood. So inulin is a very good biochemical which is used in order for you to measure the glomerular filtration rate of blood. And the answer for the glomerular filtration rate per 1.73 meters cube of body surface area. Meters cube of body surface area is going to be 120 milliliter per minute, which is comparable or as 180 liters per day. So now, in case where you have now this other part here, you have now the reabsorption of an ion and you have secretion of ion. We are no more going to speak so much about the clearance, but we are going to speak about the fractional excretion, So now we need to know that clearance of inulin is going to be the factor which is equal to the glomerular filtration rate. But now we need to know that the clearance of substance in this case is going to be due to both, so the presence of a substance inside the urine will depend on the filtration of the substance here and the reabsorption of the substance. So we see that since the substance is reabsorbed, the clearance will be lower than the glomerular filtration rate. This is why the fractional excretion here will be less than 1.0. Now in this other case, you realize that you have the clearance here, or the amount of the substance present in urine will depend on the filtration and on the secretion. We see that filtration plus secretion is greater than just filtration. So in this case, the fractional excretion will be greater than 1.0 when the substance is secreted inside the filtrate. So here we see that the clearance will be equal to the filtration plus the secretion, but in this case it was equal to the filtration minus the reabsorption. So in this case you are going to have 1, which is the glomerular filtration minus the reabsorption, so it will be less than 1. Why in this case, when it is secreted, it will be 1 plus the secretion all over glomerular filtration, which is going to be greater than 1.0. So now the next part are going to be transport processes involving in the nephron. So now let's visualize the transport processes in the nephron. So we have fractional excretion. This is still fractional excretion, and we said fractional excretion is a Cx of the substance divided by glomerular filtration rate, or the Cx of inulin, or is a clearance of the substance, or the clearance of inulin. Now when you are going to substitute this initial formula here, because this is in case of inulin, you are going to place x here and px and all substitute this, you are going to have this other formula here. So now let's visualize the tubular transport. The tubular transport are going to be separated into different parts. At first you have filtration. At this upper portion here, at the proximal convoluted tubules, you have active reabsorption here. After the proximal convoluted tubules, when you are going downward, the more because the active reabsorption is occurring because the osmotic pressure at the proximal part of the filtrate, at the proximal part of the tubules, is going to be similar to osmotic pressure inside blood. So in order to make sure that the filtrate enters inside blood, you require active reabsorption. Now when you have done active reabsorption, you know that you are going to see that water is going to be much more concentrated in here than in here. So passive reabsorption of water is going to occur. So that's the next thing. After that, you have transcellular secretion, then you have other certain secretion. So this is going to show the overview of the different reabsorption of substances inside the blood from the tubules. So at these sessions here, we have a primary active transport and a passive transport of sodium ion, calcium ion, and potassium ion. Here you have a secondary active transport. The primary active transport is utilizing directly ATP. That is why the primary active transporters are going to be directly connected with ATPases in order to break down ATP to produce energy. While secondary active transport does not require directly the use of ATP, but requires another molecule which is going to move down its concentration gradient. Here you are going to have glucose, amino acids, and all that moving via secondary active transport. So this is secondary active transport. Here you have the chloride ion, which are going to move via passive transport, and all these they are going to move passively. So in yellow here you have passive active transport. After in passive active transport, this is secretion. So you have a certain secretion here. So now this one is also having secretion. With a secretion, an active secretion, you see that there is a certain molecule here, you have active secretion, but here is only passive secretion. So now it descends here, you are going to have reabsorption of water in the descending limb. In the ascending limb, you are going to have a reabsorption of salt via the primary active transport and secondary active transport. And up here in the distal convoluted tubules, you are going to have primary active transport and secondary active transport acting. The primary active transport will be due to the sodium ion potassium ion pump, while the secondary active transport will be due to the sodium ion potassium ion importer. And here you are going to have the potassium ion secretion via a passive transport via the potassium channels. Then you are going to have here the sodium ion reabsorption all through the distal convoluted tubules. Here you are going to have the secretion of hydrogen ion, reabsorption of sodium ion, and all that. So now let's visualize the next part of this tutorial, which is going to be the different concentration of ion which are being reabsorbed and secreted at different portions. So the water, we see that in proximal convoluted tubules, 65% of water has been reabsorbed, and only 10% of water is reabsorbed inside the loop of Henle. So the total reabsorption will be 93 to 99%, and the fractional excretion. So fractional excretion, we said the fractional excretion equals to the clearance of the substance divided by the clearance of inulin. So now we see that this substance has a fractional excretion which is less than 100%, or which is less than 1, the whole fraction 1. So it implies that this substance is mostly reabsorbed. As we can see here, all of them have a fractional excretion. All of them, the sodium ion, potassium ion, calcium, and all of their fractional excretion are less than 100%, or less than, when we have 100% is equivalent to 1 whole fraction. So you have all of them are going to be less than 100%. So it implies that all of them are going to be reabsorbed inside the body. So now, when we visualize this other one, para-aminohippuric acid, we see that the fractional excretion is going to be 500. It's greater than 100%. This shows that it is the para-aminohippuric is much more secreted. As you can see here, secretion. So it shows that the fractional excretion greater than 100% is going to show you a secretion that has occurred. Now we have creatinine here. Creatinine has an exact fractional excretion of 100%. This shows you that you know that the formula of fractional excretion is clearance of creatinine all over is going to be a clearance of creatinine over the clearance of inulin. So now if it's 100%, it implies that you are going to have a value of 1. So it implies the clearance of creatinine will be equal to the clearance of inulin. And this is a reason why creatinine can be used as another measure for the glomerular filtration rate in the body. The perfect measure is made by using inulin, but creatinine can also be used as another measure in order to find the glomerular filtration rate of the body. So that's it. And the importance of the importance of creatinine is that creatinine is endogenous and it is produced continuously inside the body, while inulin has to be injected. So now let's visualize now for sodium ion. For sodium ion, normal concentration inside the blood is 153 millimoles per liter. Then you have 65% absorbed in the proximal convoluted tubule, 25% absorbed in the loop of Henle. That makes 95 to 99% reabsorbed, and the fractional excretion is only 0.5 to 5%, controlled by aldosterone, ADH, and NAP. So you have potassium, you have the value 65% here. Potassium is very low extracellularly, 4.6 inside blood. You have 65% reabsorbed, you have 10% reabsorbed, and now you see that potassium is secreted. So this is why the ranges of potassium is going to vary from 2%, a less than 100%, and even greater than 100%. It is less than 100% if the potassium has been reabsorbed, and it will be greater than 100%, going to 150% if the potassium is much more secreted and it's controlled by aldosterone. The more aldosterone is produced, the more secretion, the more fractional excretion is greater than 100%. So we have calcium ion here. Is going to 60% is going to be reabsorbed in the proximal convoluted tubules, 30% in the loop of Hindley, 95 to 99% is going to be the totally reabsorbed, and then you have 1 to 5%. This shows that calcium is not secreted. But in case where you have parathormone excess, if you have a parathormone excess, you realize that calcium ion is going to be secreted and is going to be greater than 100%. So now magnesium ion, you have fractional excretion is going to be greater than 100%. So magnesium ion, you have 15% here absorbed in the proximal convoluted tubules, while a very large amount is absorbed in the loop of Hindley, 70%. So that makes a total of 80 to 95% reabsorbed by reabsorption of magnesium ion, and we have 5 to 20% reabsorption of magnesium ion. So it means that magnesium ion is mainly reabsorbed. Chloride ion, you have 112 millimoles per liter inside blood. Then you have this one here, and lastly all this. So these are all the concentrations of the different values. For hydrogen carbon is 24 millimoles per liter, and you have the values there is going to be less than 100%. This shows that it is less, it is much more reabsorbed. But in case where you have an alkalosis, they are going to be secreted, and the value is going to be greater than 100%. For glucose, normally glucose is not present inside blood, so all the glucose is going to be reabsorbed. It's only when you have a glucose intolerance that you can have glucose inside blood. So this is why here is going to be 0. So since all is going to be reabsorbed, the creatinine the clearance of glucose will be equals to 0. So none of it will be present inside urine. So fractional excretion is going to be 0%. So now let's visualize the reabsorption. We have reabsorption of glucose and amino acid. So we need to know that the glucose is capable of being filtered because it is very small in size compared to the protein. So we have glucose filtration graph is going to be following this blue line here. Now the glucose reabsorption is going to follow the green line. So we realize here that the green line is going to show glucose reabsorption. But the problem is that when you attain a maximal plasma concentration, when you attain a plasma concentration of something like 20 millimoles per liter of glucose, you are going to have a maximal reabsorption rate which is going to occur. That is, all the carrier proteins involved in the reabsorption of the glucose are going to be full, or they are going to be enclosed, are going to be closed by excess concentration of the glucose. So this is why you are going to have a glucosuria. So in this case, when your value is still less than when you have is still less than 15 millimoles per liter, when your glucose concentration of blood is less than 15 millimoles per liter, you realize that the filtration of glucose is equivalent to the reabsorption of glucose at this position here. So this is why the graph that is going to be produced is going to be 0. There is no glucose in urine. But now when you attain something like 10 millimoles per liter of glucose inside blood, you see that this one is tending now to be equilibrating, and they feel that glucose is much more, the graph is much more steeper compared to this one. So you see that there's going to be release of glucose inside blood. So now let's visualize the general reabsorption of organic substances. The general reabsorption of organic substances is going to occur via sodium ion reabsorption as such. So you see that normally the sodium ion is reabsorbed inside intracellularly. There's going to be very low sodium ion because of the sodium ion potassium ATPase here. The sodium ion potassium ATPase causes the sodium ion to constantly flow inside the blood and reduces sodium ion concentration inside the cell. So this reduction in sodium ion concentration inside the cell makes this other portion here, the inside the lumen, to be very high in sodium ion compared to intracellularly. So now in order to reabsorb the organic substances like glucose and amino acid, phosphate, lactate, sulfate, you see that sodium ion is going to move down its concentration gradient, while these other ones are going to move up their concentration gradient. So this is going to be secondary active transport with a sympater mechanism, where the X are going to enter now into the cells as such here, and sodium also enter. Now the reabsorption of the oligocytes. So let's visualize the reabsorption of the oligopeptide. In order to reabsorb the oligopeptide and dipeptide dipeptides are reabsorbed via the hydrogen ion and the peptide syn-porter. So we have the hydrogen ion syn-porter here. So that is in the reabsorption of dipeptide. We have the hydrogen ion syn-porter involved here. And for the oligopeptide, you first have an extracellular peptidase on the proximal convoluted tubule, which are going to convert the oligopeptide into amino acid, and the amino acid are going to be reabsorbed via this mechanism we have already spoken about with the sodium ion syn-porter. So now let's visualize the reabsorption of proteins by endocytosis. So now if it's completely a protein, so the complete reabsorption of protein is going to occur by endocytosis, where you're going to have the receptors on the surface. After having the receptors, they are going to bind with the primary lysosome. The receptor is going to be recycled coming up here, and the primary lysosome is going to convert, is going to bind with the molecules which has been reabsorbed by endocytosis. Because they are very large, they cannot be converted by extracellular peptidases, so they are going to be absorbed by endocytosis, then come into this secondary lysosome. After having the secondary lysosome, we are going to have hydrogen ion secreted via the hydrogen ion ATPase or hydrogen ion pump, and then they are going to, the lysosome contain enzymes which are going to split the amino acid. They split the proteins now into amino acids. Then they are going to flow now inside the passive carrier system inside the blood. So now let's visualize now the secretion and excretion mechanism of para-aminohipurate. We need to know that the filtration of para-aminohipurate is going to follow this blue line, while the secretion of para-aminohipurate is going to follow the green line. So secretion occurs much more than filtration at this level here. So the stiffness of secretion is going to be much more than filtration. This is why para-aminohipurate is much more secreted than is being filtered. So the presence, the body does not tolerate the presence of para-aminohipurate and even certain drug inside the body. So this is why it's going to produce a resultant graph, which is going to be as such here shown in red. So you're going to have this resultant graph. So now the secretion of organic anions, example. So how does this secretion of para-aminohipurate and other organic ions occur? So it's going to occur as such. So realize that there's this particular syn-porter here. So this syn-porter, this antiporter, is going to take out the alpha ketoglutarate, which is going to be produced from the mitochondrion. It's going to take out alpha ketoglutarate, and then this is going to flow up the concentration gradient. The question is, why is it flowing up the concentration gradient and this alpha ketoglutarate is flowing down the concentration gradient? Realize that alpha ketoglutarate is negatively charged, and these anions here are also negatively charged. But you know that due to the constant activity of the sodium ion potassium ion pump, there is going to be the flow of the sodium ion, of 3 sodium ion outside the cell, and a flow of 2 potassium ion inside the cell. And also the potassium ion are going to flow again back via the potassium ion channel outside the cells. This is going to cause much more positive ion charge located in the blood, and also much more positive ion charge located in the lumen due to the action of sodium ion potassium ion ATPase. So this is why it makes the blood at level of the blood in the proximal convoluted tubules to be positively charged, and at the lumen is going to be positively charged too. But inside the cells, these two parts are going to be negatively charged. So now it will be very difficult for this negative anion to be secreted to pass to a negative charge, because it is here is going to be negative, so it's going to interact it. So in order to prevent now, in order to counteract the to counter the effect of this charge, they are going to do an antiporter. You realize that alpha ketoglutarate is negatively charged, so now it's going to flow down its electrical gradient. So alpha ketoglutarate is going to flow down its electrical gradient to go to a much more positive side, while the organic anion is going to flow now up the concentration gradient. So this is going to be secondary active transport, which is going to be an antiporter. So now in this case here, you have now how alpha ketoglutarate enters. Alpha ketoglutarate is going to enter via the transport of this protein here. This protein is making sodium ion to enter, and it's going to cause the alpha ketoglutarate to go up the concentration gradient. So now when the anions are present here, you see that they are going to flow easily out of the cell. You see that this is negative charge, this is positive charge, so you can easily flow via carrier system out of the cell, and this other conjugate can flow out of the cell via the ATPs. So now let's visualize cations. In contrary to anion, it will be easier for the cation to flow inside the cell here at the basolateral side. So you see that epinephrine, choline, histamine, and all this. In this case of anion, you have paraminohippurate, you have phenol red, hippurate, urate, and all that, paraminohippuric acids and all that, with penicillins. All of them are negative anions. Here you have positive cations, you have epinephrine. They are going to flow directly inside via carrier proteins, and then outside is going to flow up the concentration gradient, because here is positive, so it's going to flow up their electrical gradient, and then the hydrogen ion is going to flow down its electrical gradient. So this is a secondary active mechanism with a hydrogen antiporter, hydrogen ion antiporter, and it can also flow up here by an ATPase, by primary active transport, which is associated with the ATP. So now let's visualize how the electrochemical gradient of sodium. So we I've initially told you that you have the potassium, sodium ion potassium ion channel, which is constantly in activity at the basolateral side of the different cells in the convoluted tubules and loop of Henle. So now you are going to have this is going to cause the concentration inside the cells of sodium ion to be very low, and concentration of sodium ion outside the cell to be very high. Also, you realize that the potassium ion, as it's going to enter, is also going to flow out here, and it's going to flow out here. So it's going to maintain a negatively charged inside the cell. So you are going to have both the concentration gradient of sodium ion outside the cell is going to be high, and the positive charge outside the cell is going to be high too. So this is going to cause a chemical driving force of sodium ion and an electrical driving force of sodium ion. This is why sodium ion constantly flows inside the cells, and sodium ion is going to be associated with many symporters like the cation symport and the anion symport. So now let's visualize the sodium ion and chloride ion reabsorption. So how is sodium ion and chloride ions reabsorbed? So we have the electroneutral sodium ion hydrogen ion exchanger. So at first you know that at this position here, you're going to have the sodium ion ATPase, which is going to make sodium ion move up the concentration gradient, reduce the concentration of sodium ion here. So what happens is that the sodium ion is going to flow inside down the concentration gradient, while hydrogen is going to move up the concentration gradient into the proximal convoluted. So at early proximal convoluted tubule, sodium ion is going to flow down the concentration gradient inside the cell, and then hydrogen is going to move up the concentration gradient. So also the sodium ion is high in concentration out inside the lumen than inside the cell, so it's going to flow down the concentration gradient, and also going to cause the reabsorption of glucose, and glucose are going to flow via their channels and their carrier in the basolateral side. So all these are going to cause now when all this flow of sodium ion is going to cause this extracellular part here in the lumen to be much more negative charged due to the presence of chloride ion, and here will be much more positively charged. So this is going to cause the chloride ion reabsorption via a paracellular pathway. And also after all this movement of the ions, you are also going to have a water reabsorption, because this is becoming much more concentration. So water is going to move via here is going to be paracellular pathway, and water is going to move here via a transcellular pathway. So this is also going to create again inside the mid and late proximal convoluted tubules. It's going to create again a much more chloride ion concentration here. So it's going to flow again through the paracellular pathway, and this is going to produce again another positive charge here, which is going to cause the different positive ion to flow through the electrical pathway toward the blood. So it's going to flow now in a paracellular pathway into the blood. So you have potassium, sodium, and calcium ion due to this electrical gradient here. They're going to flow in the paracellular pathway into the blood. So now let's visualize in a thick ascending limb of the loop of Hindley. So this was in the we know that in this position here, descending limb, you know that you are going to have only the reabsorption of water. Now in the ascending limb, what do we have? We have the reabsorption of iron. So how does the reabsorption of iron occur in the ascending limb of the loop of Hindley? The ascending loop of Hindley is much more impermeable to pure water but permeable to the ions. So what's going to happen is that you are going to have first this sodium ion, 2 chloride ion, potassium ion, syn-porter. So this syn-porter is going to move the sodium ion down its concentration gradient. Potassium is going to move across an electroneutral gradient, and chloride ion is going to move up the concentration gradient. Whereas the chloride ion is going to move out of the cell. So you have 2 anions are going to move out of the cell, while only 1 sodium ion is moving out of the cell here. So you have 2 chloride ion and only 1 sodium ion moving out of the cell, and realize that the potassium ion which has initially entered is going to move back into the lumen. So we realize that you are going to have a resultant change. At this position, you are going to have a much more negative, because there are 2 chloride ion with only 1 potassium ion here. So this makes this position to be negative, and you see that the potassium ion which had entered is going to flow back out, and this is going to make this position to be positive. So it's going to create a positive negative gradient, a positive negative gradient here. So the positive ions on this side are going to flow inside via the paracellular pathway. So this positive ion are potassium, sodium, and calcium, and magnesium ion. Now the distal convoluted tubules, you have sodium ion, chloride ion, syn-porter. So the sodium ion is going to flow down the concentration gradient. The concentration gradient of sodium ion is going to be maintained by the ATPase, the sodium ion, potassium, syn-porter, and the chloride is going to move out via the chloride ion channels. So here you are going to have the sodium ion transported via the channels. You have this one is going to move down the inside the collecting duct. They are going to move down the concentration gradient, and then here is going to be maintained. They are going to move up the concentration gradient via the ATPase. So now let's visualize the countercurrent system. So now with the countercurrent system, if you have a temperature of 0 degrees here and you have a temperature of 100 degrees here, all of them are going to be having a temperature of 50 degrees. So now if we exchange the countercurrent, in case here while here is 100 degrees and they are going here, so you see that this temperature is going to take a resultant temperature and decrease this by 10. So here will be 90 degrees, it will be closer to this temperature, and this will be closer to this temperature, here will be 10 degrees. So this is what is involved mainly in the process of countercurrent mechanism. With the countercurrent mechanism, what you realize is that the descending limb is going to be permeable to water, while the ascending limb is going to be permeable only to salt. So you realize that the more you are descending inside the loop of Hindley, you realize that the concentration inside the lumen is going to increase, while the concentration outside here is going to decrease, because water is flowing outside. So we have the concentration inside the lumen is going to decrease inside the countercurrent mechanism, and then as it's going to move up here, the concentration is going to increase. So water, you need to know that the concentration outside here is going to be high first. It's going to be something like 600. It's only permeable to water. Water is going to flow only outward. It's only permeable to water, and here upward here they are going to have active transport mechanism which are only permeable to ions. So this is the countercurrent mechanism, and the countercurrent mechanism, realize that all the concentration at each level are going to be similar. So the concentration here is going to be 400, 400, 400. Here is 600. Here all going to 1000. Here is 1200 at this lower portion here. So we have 800 here, because the ions, the more the ions are moved here by active transport. So you realize that at this portion you are going to have 800, 200. But here is still going to be 400 because they are removed by active transport. So now we have water reabsorption and excretion. So we see here that as the plasma enters, we have glomerular filtration rate of water is completely 100%. So 100% of water is going to flow. You have 65% going to be reabsorbed with sodium chloride. Here you are going to have the rest here of 35%. So the 35% are also going to be reabsorbed now inside the loop of Henle, and they're going to move up here. You see that water is going to be slightly secreted. So you're going to have side secretion. So you're going to have side secretion, so 25% here. So this is why it's going to increase by 25%, and water here is going to be reabsorbed again much more again inside the collecting duct. So that's why you have 0.5 to 5% of water which are remaining. So now let's visualize how urea is going to maintain the osmolarity of the medulla of the renal of the kidney. So now it starts first with urea here passing in through. You are going to have a concentration of 5 millimoles or 100% of urea at this level here. Passive reabsorption of urea is going to occur. You are going to remain with 50% of urea here, and after you need to know that urea is going to reabsorb at the collecting duct and is going to enter now into the interstitium of the medulla in order to maintain the concentration that we have seen above there. So the concentration of the countercurrent mechanism is going to be maintained by urea. So this is why urea is going to be reabsorbed at the level of collecting duct. So you have 50% here is going to descend and then going to ascend. No urea is going to be absorbed in the distal convoluted tubule and then the upper limb of the loop of Henle. None of them is going to be absorbed there, but it's going to be reabsorbed back at the collecting duct. So you see that this upper portion, it is not supposed to increase in concentration. This is why there is no reabsorption of urea in this position. It's only the lower portion, which is at the level of 1200 millimole per liter, that needs to be increasing concentration. This is why only urea is absorbed at the level of the medulla, but not at the level of the cortex here. So now let's visualize water deficit. So this is the general part of the body involved with water homeostasis. So from here we've seen the first section of the kidney, and now we can now pass to the second section of the renal physiology. We say thanks for your kind attention. Please don't forget to like and subscribe for Science Makers. Thank you.